What if a drug could be, programmed like software, like just writing code to silence a disease gene?
Right, just typing it out.
Yeah. Or what if we could stop harmful proteins before they're even made, before the body even builds them?
Which is completely wild to think about.
It really is. I mean, what if some of the most difficult genetic diseases on the planet could be treated by targeting RNA directly?
Which is exactly what we're getting into today.
We're going straight into the deep end on this one because we're exploring a class of medicines that, honestly, it sounds like pure science fiction.
It really does. But it is very real.
Right, and it's rapidly rewriting the rules of modern medicine. We are talking about antisense oligonucleotides,
Sorry, ASOs.
ASOs. In the simplest terms, these are short synthetic strands of nucleic acids, and they're designed to bind to matching RNA in your cells and then completely alter what happens next.
Yeah, the paradigm shift here is, I mean, it's hard to overstate. And to ground this conversation for you, we are relying on a massive stack of scientific literature today.
Oh, a ton of sources for this deep dive.
Definitely. We're looking at structural activity relationship studies, rigorous safety assessment protocols.
The heavy chemistry stuff.
Right, the heavy chemistry. And historical reviews tracking how this whole technology clawed its way from just a theoretical concept in a lab to actual clinical applications.
Decades of work.
Decades of medicinal chemistry and molecular biology.
So our mission for this deep dive is to trace that exact journey. We are going to explore how ASOs evolved from this bold idea into a powerful, programmable therapeutic platform, one that can treat diseases that the medical community once thought were just hopelessly, completely undruggable.
Undruggable, yeah.
That was a word. But before we get into the solution, we really have to deeply understand the fundamental problem. Like, why did we even need to invent programmable RNA drugs in the first place?
Right.
Because if you look at traditional medicine, I mean, we've done pretty well with pills and injections for the last century.
We have, yeah. But we've been operating with this massive blind spot.
Okay, a blind spot.
Yeah. And to see it, we really need to look at the central dogma of biology.
Right, the core flow of information.
Exactly. The core flow of genetic information in your cells. It goes from DNA to RNA to protein.
DNA to RNA to protein.
Right. So think of your DNA as this master reference library, locked away in the nucleus of your cell.
Deep in the vault.
Exactly. It's pristine, it's protected, and it contains the instructions for literally everything your body will ever need to build.
And when your cell actually needs to build something like, say, a new muscle fiber or an enzyme to digest your lunch, it doesn't take the master blueprint out of the vault, right?
No, that would be way too risky.
It makes a working copy, a disposable photocopy.
Right, that's the messenger RNA, the mRNA.
Okay.
And that mRNA travels out of the secure nucleus and into the messy cytoplasm. And that's where the cellular machinery reads it and uses those instructions to build a protein.
And the proteins are the actual, physical machines doing the heavy lifting in your body.
Exactly, they do the work, which means if there's a mutation in your DNA,
the master copy.
Right. That mutation gets dutifully copied right into the mRNA.
It photocopies the typo.
Yeah, the typo goes right along with it. And then the cellular machinery reads those flawed instructions and builds a broken, malfunctioning, or even actively toxic protein.
Wow. Okay.
And for over a century, drug development has been almost entirely obsessed with fixing or blocking those broken proteins.
So we've been completely focused on the end product. The end of the assembly line.
Entirely. But the scientific literature highlights this really staggering reality about our genome.
What's that?
Only about 1.5% of human DNA actually encodes for proteins.
Which, that always blows my mind, 1.5%.
It's tiny.
I mean, if I buy a massive encyclopedia set and only 1.5 pages actually have text on them, I'm going to feel completely ripped off. What is the rest of the DNA doing?
Right. Well, for a long time, scientists casually dismissed the rest as just junk DNA.
Junk DNA, yeah, I remember that term.
Yeah, they assumed it was just evolutionary baggage. But our understanding has radically shifted now.
Because it's not junk.
No, not at all. We now know that the vast majority of that so-called junk is actually made of these highly structured, functionally critical regulatory elements.
Yeah.
And non-coding RNAs.
So it's management.
Exactly. It's the management system. It tells that 1.5% when to turn on, when to turn off, and how loud to be.
But traditionally, the pharmaceutical industry has just ignored the management system.
Completely ignored it. They focused all their firepower on the workers, the proteins.
Right.
So conventional drugs like small molecules, think of aspirin or statins or large biologics like monoclonal antibodies, They're designed to bind directly to fully formed proteins.
So the traditional approach is to look at the broken 3D protein machine, right, and find a little pocket or a groove or a crevice on its surface.
Yeah.
And then you design a chemical key that slots perfectly into that pocket to just jam the gears.
That is the standard playbook, yeah.
Yeah.
But here is where the industry hits a massive brick wall.
Okay.
Up to 80% of those protein coding targets are considered undruggable by conventional methods.
Wait, 80%? That is a massive failure rate for traditional medicine. Why are so many proteins immune to our drugs?
It comes down to structural biophysics, really.
Right.
Many proteins are essentially flat.
Flat.
Yeah, they don't have those convenient, deep little pockets for a small molecule drug to slot into.
Yo, interesting.
All right, so they might be large, smooth structural proteins,
Yeah.
or they might function by forming these massive flat complexes with other proteins.
And a small molecule drug just, slides right off?
Just slides right off, nothing to grab onto. And on the flip side, large biologic drugs, like those antibodies, they are great at binding to flat surfaces.
Okay, so use those.
Well, the problem is they are physically massive. They cannot cross the cell membrane.
Oh, I said.
So if your problematic protein is buried deep inside the cell or inside the nucleus, an antibody can never reach it.
Wow. This paints a pretty bleak picture, honestly. We have this massive landscape of genetic diseases caused by malfunctioning proteins. And for 80% of them, traditional pharmaceutical science essentially shrugs and says, sorry, we don't have a tool that fits that shape.
Which is a staggering bottleneck for medicine.
It really is.
And that is exactly the bottleneck that ASOs are designed to bypass.
Okay, so let's look at what these molecules actually are. An ASO is a short, synthetic, single-stranded nucleic acid.
Right?
And we're typically looking at a length of just 12 to 30 nucleotides.
Just a tiny snippet of code.
Just a short string of genetic letters, A, C, T, G, or U.
Exactly. And the mechanism fundamentally shifts the paradigm. Unlike traditional drugs that have to kind of chemically grope around trying to find a 3D pocket on a fully baked protein.
Which we know 80% of them don't even have.
Right. ASOs work entirely upstream. They operate at the RNA level. They intercept the message before the protein is ever even constructed.
I love visualizing it this way. So if a mutant gene is a terrible recipe and the toxic protein is the ruin inedible cake, traditional drugs are basically trying to fix the cake after it's already baked, right? Like it's frosted and sitting on the kitchen counter, and you're trying to scrape the bad parts out without destroying the whole thing.
Which is almost impossible.
Exactly. But ASOs act like a red marker, just crossing out the bad instructions on the recipe card before the chef even turns on the oven.
That is a highly accurate way to look at it. Because they target RNA, ASOs are entirely programmable.
Programmable.
Yeah. Traditional drug discovery is often just a numbers game. You use high-throughput screening to test millions of random chemical compounds against a protein, and you just cross your fingers that one magically happens to fit the shape.
Which is incredibly inefficient.
Extremely.
Okay.
But with an ASO, if you know the genetic sequence of a disease-causing RNA, the exact string of letters, you simply design a complementary sequence that binds to it perfectly.
Because RNA operates on the most basic fundamental rules of biology, right?
Yeah.
Watson-Crick base pairing. Adenine binds to uracil. Cytosine binds to guanine. It's a totally predictable matching game. You read the bad code, and you type out the mirror image.
And you make the undruggable target entirely irrelevant.
Wow.
Yeah, if you intercept the mRNA messenger, you never have to worry about the complex folded 3D shape of the resulting protein. The protein simply ceases to exist, or it's altered before it's born.
But I have to stop you there, because if it's really that simple, literally just reading the genetic code, figuring out the mutation, and typing out the opposite sequence on a synthesizer, why are we only seeing this explosion of therapies right now?
That's the big question.
Because we've known about RNA and base pairing for a very long time. Why wasn't this the standard of care decades ago?
Well, the truth is the idea is decades old.
Yeah.
The biological theory was incredibly elegant, but the physical reality of actually putting these molecules into a human body was a nightmare.
Oh, really?
Yeah. The history of bringing this programmable concept into the clinic is one of the most turbulent roller coasters in modern science.
Take us back to the beginning. Where did the initial spark come from?
We can trace the genesis back to 1978. Researchers were working with the sarcoma virus.
Okay, 78.
Yeah, and they synthesized a tiny 13-nucleotide long strand of DNA, a 13-mer.
Just 13 letters.
Right, and they designed it to be perfectly complementary to a highly specific, conserved sequence in the virus's RNA. And when they introduced this synthetic strand into chick embryo fibroblasts that were infected with the virus, the results were absolutely stunning.
What happened?
The synthetic DNA selectively blocked viral translation and stopped the virus from replicating.
1978, we're talking almost 50 years ago.
Yeah.
The hype must have been astronomical. Imagine proving that you can just synthesize a short, cheap piece of DNA and stop a virus dead in its tracks. It sounds like they discovered the ultimate cheat code for biology.
The scientific community certainly thought they had. I mean, the initial excitement was unbounded. It felt like the dawn of plug-and-play medicine.
Sequence the pathogen, print the opposite sequence, cure the disease.
Exactly. It was viewed as a universal platform.
Mm.
But of course, biology is never that compliant.
Right.
As the field pushed forward into the 1990s and early 2000s, it hit a devastating wall. This era is often referred to in the literature as the trough of disillusionment.
The trough of disillusionment, because it turns out the human body is vastly more complex and hostile than a chick embryo cell in a petri dish.
Vastly more hostile.
If I think about this from an evolutionary perspective, our bodies have spent billions of years developing highly aggressive defenses against random floating strands of foreign genetic material.
Yeah.
If I injected a raw strand of synthetic DNA into your arm right now, your immune system wouldn't see a cure, it would see an invading pathogen.
And it would react with overwhelming force. When early developers tried to put unmodified natural DNA oligonucleotides into humans, they encountered two massive biological barriers.
Okay, what was the first one?
The first was degradation. Your blood and tissues are literally packed with enzymes called nucleases.
Nucleates, okay.
Yeah, and their entire biological purpose is to act like molecular wood chippers. They are constantly scanning for loose DNA or RNA, and they just chew it up into individual nucleotides to recycle them.
So the drug is being shredded before it even reaches the target tissue.
Instantaneously. Unmodified ASOs had a half-life in the blood measured in minutes.
Minutes?
Yeah, they were cleared out before they could achieve any therapeutic effect. But the second barrier was even worse.
Worse than being shredded in minutes.
Yes, because the small percentage of the drug that did survive triggered catastrophic immune alarms.
Wow. What does that actually look like for a patient in an early clinical trial?
Severe dose-limiting toxicity. We are talking massive flu-like symptoms, high fevers, rigors, chills, and really dangerous drops in blood pressure.
Oh my God.
The body just mounted a full-scale inflammatory response against the drug.
But why does a simple string of A's, C's, T's, and G's trigger such a violent reaction?
It comes down to a specific molecular pattern. Bacterial and viral DNA frequently contains a specific two-letter sequence, a cytosine, right next to a guanine.
Okay, C next to G.
It's known as a CpG motif, and more importantly, in bacteria and viruses, these CpG motifs are unmethylated. They lack a specific chemical tag.
So they're bare.
Exactly. Human DNA also has CpG motifs, but ours are naturally tagged with a methyl group.
So our immune system uses that tiny chemical tag to tell self from non-self.
Exactly. We have specialized immune sensors, specifically toll-like receptor 9, or TLR9, and their entire job is to scan for unmethylated CpG motifs.
Well, when it finds one.
When an early ASO happened to include a C next to a G to match the target RNA, it essentially rang the TLR9 alarm bell.
The immune system dumps inflammatory cytokines into the bloodstream, assuming a massive bacterial infection is underway.
Right, so you have a drug that is either completely destroyed in 5 minutes, or it survives just long enough to put the patient in the hospital with a systemic inflammatory response.
That is brutal.
The clinical failures really piled up. I mean, there was one early drug approved for a specific eye infection in the late 90s, but it required direct, localized injection into the eye.
Yikes.
Yeah, and it was eventually withdrawn when better treatments came along. Other high-profile, late-stage clinical trials, especially in oncology, they completely failed to show efficacy. The drug couldn't get to the tumors in high enough concentrations, or the patients just couldn't tolerate the doses.
So the whole field stalled.
By the early 2000s, investors fled, pharmaceutical companies shut down their RNA programs entirely, and many considered antisense technology a total dead end.
It's a classic example of having the right biological map, but just the wrong vehicle.
Exactly.
The matching game of Watson-Crick base pairing was totally sound, but the chemistry was fundamentally not ready for prime time.
And the pivot that saved the entire field wasn't a biological discovery. It was a grueling, decades-long marathon of chemical engineering. The modern ASO is a marvel of synthetic chemistry. Chemists painstakingly tweaked individual atoms on the sugar phosphate backbone of DNA to armor these molecules.
They had to build molecular armor.
They had to disguise them from the immune system, harden them against the nucleuses, and ensure they could survive the journey to the interior of the cell.
Okay, so let's follow that journey. Let's assume we have a modern chemically armored ASO. It gets injected into the bloodstream, it survives the nucleuses, it ignores the immune alarms, and it arrives at the target cell.
Right.
It still has to get inside to find the RNA, right? How does a synthetic strand of DNA breach the cell membrane?
It relies heavily on a process called endocytosis. The cell membrane essentially folds inward, engulfing the ASO and bringing it inside, trapped within a small cellular vesicle called an endosome.
I always think of the endosome as a cellular stomach.
That's a good way to put it.
Like the cell takes in this material from the outside, wraps it in a membrane, and starts pumping acid into it to digest whatever's inside.
The cellular stomach analogy is incredibly apt because that acidic digestion is exactly what the ASO has to avoid. Endosomal escape is one of the most critical and challenging bottlenecks in all of RNA therapeutics.
Because it has to get out before it's digested.
Exactly. As the endosome matures and becomes more acidic, the ASO has to physically destabilize that membrane and break out into the cytoplasm.
It has to pull off an escape room sequence while the walls are literally dissolving it.
And honestly, it's highly inefficient. The literature suggests that only a tiny fraction, sometimes 1% or 2% of the ASOs taken up by the cell actually manage to escape the endosome.
1 or 2%.
The rest are trapped and eventually degraded. But because ASOs are so potent, that 1% is often enough.
Okay, so our 1% of survivors break out of the endosome. They're now floating in the cytoplasm, or perhaps they traffic into the nucleus.
Right.
They scan the massive sea of RNA until they find their perfect complementary Watson-Crick match, and they bind to it.
Yes.
What happens next? How does this actually stop the disease?
ASOs fundamentally rely on two primary mechanisms of action to neutralize the threat. The first is degradation. Specifically, RNase H1-mediated degradation.
RNase H1. Okay, let's unpack the mechanics of this enzyme.
RNase H1 is an endogenous enzyme, meaning it's a tool your cells naturally produce and maintain.
It's already there.
Right. Its evolutionary purpose is to patrol the cell, looking for a very specific, highly unusual structure, a long strand of DNA bound perfectly to a long strand of RNA.
An RNA-DNA hybrid. But why is that unusual? Doesn't DNA make RNA all the time?
It does, but those interactions are fleeting. A stable, long DNA-RNA duplex floating in the cytoplasm usually signals a viral infection like a retrovirus trying to integrate, or a catastrophic mistake in cellular replication.
So the cell sees it as a major red flag.
Exactly. RNase H1 acts as a molecular security guard. When your synthetic ASO, which is built on a DNA-like chemical scaffold, binds tightly to the target messenger RNA, it forms exactly this unnatural hybrid.
Ah, the ASO is basically painting a massive target on the back of the disease RNA.
You got it.
It binds to it, creates the forbidden shape, and just waits for the security guard to arrive.
Precisely. RNase H1 recognizes the hybrid geometry. It uses a magnesium ion as a critical cofactor to power its catalytic core, and it locks onto the hybrid and precisely cleaves the RNA strand right down the middle.
It just cuts the recipe right in half.
It shreds the messenger RNA. But the true brilliance of this mechanism is its asymmetry.
What do you mean?
RNase H1 only cleaves the RNA strand. The synthetic ASO is completely untouched.
Oh.
Once the RNA is cut, it falls away to be fully digested by the cell. And the ASO is then completely free to hunt down another copy of the target mRNA and repeat the process.
It's catalytic.
Exactly.
One single ASO molecule can take out multiple bad recipes day after day.
Yes.
That totally explains why that tiny 1% that escapes the endosome can still be so incredibly effective.
That's right.
Okay, so that covers the search and destroy mechanism. What's the second way they work?
The second major mechanism is entirely different. It's called steric blocking, or splice switching. And in this scenario, the ASO doesn't trigger any enzymes to destroy anything.
Okay.
Instead, it acts as an immovable physical roadblock.
A roadblock. Let's set the stage for where this happens.
This typically happens inside the nucleus, targeting pre-mRNA. When your DNA is first copied, the raw transcript is called pre-mRNA. It's rough, unedited material.
Right.
It contains the actual coding instructions, called exons, but they are broken up by long stretches of non-coding gibberish called introns.
It's like a raw film reel. The good scenes are the exons, but in between every scene is 5 minutes of the camera pointing at the floor. The introns.
A perfect analogy. Your cell has a massive molecular machine called the spliceosome. It acts as the film editor.
Okay.
It scans the pre-mRNA, identifies the boundaries between the introns and exons, cuts out all the useless floor footage, and meticulously stitches the good scenes, the exons, together to make the final, clean, mature mRNA that can be translated into a protein.
And the ASO interferes with this editing room.
It hijacks the editing process. By designing an ASO to bind to incredibly specific locations, like the exact boundary where an intron meets an exon, the ASO physically masks that splice site. It just sits there, blocking the spliceosome's access.
So the film editor can't see the cut mark.
Exactly.
So what does the editor do?
It just skips over it and moves to the next mark.
Oh, wow.
And you can use this to manipulate the final transcript in profound ways. If there is a disease-causing mutation inside a specific exon, you can use a steric-blocking ASO to hide that exon from the spliceosome. The cell edits it out entirely, and you've bypassed the mutation.
That is wild.
Or, conversely, if the cell is mistakenly leaving an important exon out, you can block the inhibitory signals that cause the skipping, forcing the spliceosome to include the necessary sequence.
That is genuinely staggering. We aren't just breaking things. We are actively tricking the cell's editing machinery into rewriting the genetic software on the fly.
It is incredibly powerful.
And the literature notes this roadblock technique isn't just for splicing, right?
Correct. You can use steric blocking in the cytoplasm as well. If an ASO binds to the mature mRNA, right at the starting line, the 5' cap, where the ribosome normally attaches to begin building the protein, it physically blocks the ribosome from ever boarding.
So no RNA is destroyed, but no protein is translated.
Right. It's a translational blockade.
Okay, we've outlined the core biology. We have RNase H1 shredding RNA, and we have steric blocking hijacking the spliceosome. Let's connect these abstract molecular mechanisms to actual human suffering.
Okay.
How does knocking down an RNA transcript translate to stopping a toxic protein in a patient?
The most direct and dramatic application of the RNase H1 mechanism is in dominant genetic diseases, particularly toxic gain-of-function disorders.
The phrase toxic gain-of-function sounds inherently ominous. It implies the protein isn't just broken, it's actively malicious.
That's exactly the distinction. In a loss-of-function disease, a mutation causes a protein to stop working. The factory line stops. But in a toxic gain-of-function disorder, the mutated gene produces a protein that actively damages the cell.
It takes on a new destructive role.
Yes. Huntington's disease is the classic example. The mutant Huntington protein misfolds, clumps together into these massive toxic aggregates, and literally physically chokes the neurons to death.
So in Huntington's, you don't need to fix the protein, you need to shut off the faucet. You need to stop production entirely before the aggregates can even form.
Yes. By delivering an ASO that binds to the mutant Huntington mRNA, you trigger RNase H1 to shred those transcripts. You drastically lower the production rate of the toxic protein, allowing the cell's natural cleanup mechanisms to clear away the existing aggregates.
You relieve the toxic burden.
You do. But designing this drug introduces one of the most complex challenges in the field.
Which is what?
The healthy copy of the gene.
Oh, right.
In dominant genetic diseases, you usually inherit one mutated copy of the gene from one parent and one perfectly healthy wild-type copy from the other. Your cells are producing both the toxic protein and the healthy protein.
And if the healthy protein is doing an essential job, you can't just wipe out all production of that protein type without killing the patient.
Exactly. This is the difference between non-allele-specific knockdown and allele-specific knockdown. If your ASO targets a sequence of the mRNA that is shared by both the healthy and the mutated versions, it will shred both.
Which is bad.
In some diseases, the body can tolerate a 50% drop in the healthy protein without major issues. But in other diseases, the wild-type protein is absolutely vital for cellular survival.
But if the sequence is mostly identical, how on earth do you tell the ASO to ignore the healthy copy and only attack the mutated one?
You leverage the extreme precision of Watson-Crick base pairing to perform allele-specific targeting. Often, the difference between the devastating mutant gene and the healthy gene is a single letter change in the genetic code.
Just one letter.
A single nucleotide polymorphism, or SNP.
One single letter difference in a transcript that might be thousands of letters long.
One letter. And chemists designed the ASO to center perfectly on that single mutated letter. When the ASO encounters the mutated RNA, it forms a 100% perfect match, triggering RNase H1 to destroy it.
But what about the healthy RNA?
When that same ASO encounters the healthy RNA, there is a one-letter mismatch right in the middle of the hybrid.
It bubbles. They don't line up perfectly.
Exactly. That tiny single nucleotide bulge destabilizes the complex just enough that RNase H1 refuses to cut it.
Wow.
The ASO falls off, leaving the healthy RNA intact. It acts as a molecular sniper, hunting down the mutant transcript with unbelievable selectivity while preserving the wild type.
That level of precision feels almost magical. But, I mean, to pull off a sniper shot like that inside a living organism, we have to talk about the armor. We mentioned earlier that natural DNA gets shredded in minutes and triggers massive immune reactions.
Right.
To make these mechanisms work, chemists had to completely reinvent the molecule. Let's dig into the chemistry of survival. What does molecular armor actually look like?
It looks like manipulating the fundamental atomic structure of the nucleic acid. The molecular design of ASOs is a masterclass in medicinal chemistry. The modifications fall into several distinct zones. Let's start with the foundation, the backbone.
The structural scaffolding that holds the A, C, T, and G letters in a row. A natural DNA backbone is made of alternating sugar and phosphate groups, right?
Correct. In a natural phosphodiester backbone, each phosphate group has a non-bridging oxygen atom. The first monumental breakthrough in ASO chemistry happened when scientists discovered they could replace that single oxygen atom with a sulfur atom.
Okay, sulfur.
This created a phosphorothioate, or PS, linkage.
Swapping oxygen for sulfur, I mean, they are right next to each other on the periodic table, so they share properties, but sulfur is larger. What does that one atom swap actually achieve?
It changes the biophysics of the molecule entirely. First, it solves the nucleus problem. Right. Those molecular wood chippers in the blood evolved to recognize the precise shape and charge of an oxygen-based phosphate bond. When they encounter the larger sulfur atom, they can't grip the backbone properly.
So they just bounce off.
The PS linkage makes the ASO highly resistant to enzymatic degradation. The half-life in blood goes from minutes to days.
So the armor works, the nucleuses can't eat it.
But the sulfur swap does something else, arguably just as important. It makes the backbone slightly more lipophilic, meaning it has a slight affinity for fats and proteins.
Okay.
When a PS-modified ASO enters the bloodstream, it immediately sticks to circulating blood proteins, primarily albumin.
But wait, why do we want our highly engineered drug getting stuck to random blood proteins?
To avoid the kidneys. Your kidneys are exceptionally good at filtering out small, free-floating molecules from the blood and dumping them into the urine. If the ASO didn't stick to anything, you would pee out the entire dose within an hour.
Oh, I see.
By piggybacking on massive proteins like albumin, the ASO effectively becomes too large to be filtered by the kidneys. It circulates in the blood for hours or days, buying it the necessary time to seep into the target tissues and find the cells it needs to enter.
The sulfur swap is brilliant. It stops the drug from being eaten, and it stops it from being flushed away. But the literature also highlights modifications to the sugar ring itself, not just the phosphate backbone. What are we doing to the sugars?
The ribose sugar ring is where chemists tune the stickiness of the drug. Specifically, they focus on the 2' carbon position of the sugar ring. If you attach different chemical groups to that specific carbon, you alter how the ASO behaves when it binds to its target RNA.
Got it.
Two of the most critical modifications are 2'-methoxyethyl, usually abbreviated as 2'-MOE, and locked nucleic acids, or LNA.
Let's talk about LNAs, because locked nucleic acid sounds intense. What are we locking?
We are literally locking the physical geometry of the sugar ring. In an LNA, chemists build a chemical bridge, a methylene bridge, connecting the 2' oxygen atom to the 4' carbon atom of the same ring.
It's like putting a physical crossbar across the molecule.
Exactly. Natural sugar rings are slightly flexible. They pucker and shift between different shapes. When an ASO binds to RNA, it takes energy to force the sugars into the correct conforming shape. The LNA bridge locks the sugar permanently into the ideal 3D conformation for binding to RNA.
So it's pre-shaped.
Because it's pre-organized, the binding affinity skyrockets.
A rigid key fits into a lock far more powerfully than a floppy one.
The increase in affinity is massive. Every single LNA nucleotide you out to an ASO sequence raises its melting temperature by up to 5 degrees Celsius.
Let's clarify melting temperature for a second. We aren't actually melting it with heat, right?
All right. Melting temperature is a thermodynamic measurement of binding strength. It's the temperature at which half of the ASO strands unbind for their target RNA. A higher melting temperature means the ASO holds onto the RNA with incredible force. LNAs stick to their targets like molecular superglue.
Which sounds perfect. You want the drug to lock onto the mutant RNA and never let go.
You do, but there is a breaking point. High affinity comes with a severe dark side. LNAs can be incredibly toxic.
Toxic? How?
Because they are so incredibly sticky, they lose their selectivity. They start binding to RNAs that only partially match the sequence, destroying healthy transcripts.
Oh, no.
Even worse, The rigid, sticky structure can cause them to physically bind to vital cellular proteins instead of RNA, causing massive cascading organ toxicity.
So they built a drug that was too strong? How do you walk that back?
You find compromise. Medicinal chemists evolved the LNA into a new structure called constrained ethyl, or CEA. It's a slight variation on that methylene bridge. It maintains the incredible binding affinity of the LNA, but the altered shape drastically drops the toxicity profile.
It's a Goldilocks situation.
Exactly. Finding the precise Goldilocks zone of binding strength, strong enough to work, gentle enough not to rip the cell apart.
Okay, I have to pause and look at the whole picture here. We have a sulfur backbone. We have heavily modified, locked sugar rings that drastically change the shape of the molecule.
Yes.
Earlier, you told me that the RNase H1 enzyme is highly specific. It only shreds RNA if it sees a natural DNA-RNA hybrid. If we have modified the ASO so heavily that it doesn't even look like DNA anymore, won't the security guard just ignore it?
That was one of the biggest crises in the development of ASOs. Fully modified sequences, packed with 2' MOE or LNA, completely fail to trigger RNase H1. The enzyme doesn't recognize them.
So they don't work.
So the field had to invent a structural compromise known as the gapmer architecture.
The gapmer. The literature compares it to an Oreo cookie. Let's hear the cookie analogy.
The architecture is broken into 3 distinct zones. You have the 2 ends of the ASO, the wings, or the chocolate cookies. These wings are made entirely of heavily modified nucleotides, like those high-affinity 2' MOE or CH sugars.
Okay.
The wings serve 2 purposes. They resist nucleases, and they grab onto the target RNA with massive force, anchoring the ASO in place.
The cookies do the grabbing. What about the cream filling?
The center of the ASO, the gap between the wings, is a continuous stretch of standard, unmodified DNA, usually about 8 to 10 nucleotides long.
Ah, the gap is the bait.
Precisely. The modified wings latch onto the target, holding the sequence perfectly in place. The RNA bound to that central gap of standard DNA forms a classic natural DNA-RNA hybrid.
And the security guard spots it?
The RNase H1 enzyme scans along, ignores the heavily modified wings, spots the central DNA gap, locks on, and cleaves the RNA right in the middle. The gapmer architecture elegantly solved the paradox of needing both extreme stability and enzymatic recognition in the same molecule.
It's a brilliant piece of molecular engineering. But what about the immune system? We talked about how unmethylated CpG motifs trigger the TLR9 alarms, causing those massive flu-like inflammatory responses. How does chemistry solve that?
By using molecular camouflage. If an ASO sequence requires a cytosine next to a guanine to properly match the target RNA, chemists don't use standard cytosine. They synthesize a 5-methylcytosine.
A disguised version.
They artificially attach the methyl group that the human immune system expects to see. When the TLR9 receptors scan the ASO, they see the methyl group registered as human and ignore it. The immune alarms remain silent.
Okay, I want to push back on the fundamental premise of these modifications. If the sulfur backbone, the PS linkage, is so effective at stopping nucleases but causes the drug to stick to random proteins, which can cause toxicity, why even use the natural sugar phosphate scaffold at all? Why not build the drug out of something completely alien that the body has no mechanisms to break down or interact with?
That is a very sharp deduction. And it's exactly the path that led to a totally distinct class of ASOs known as PMOs, or phosphorodiamidate morpholino oligonucleotides.
PMOs? How alien are they?
Entirely synthetic. With a PMO, chemists throw away their ribose sugar ring completely and replace it with a 6-sided morpholine ring. And they replace the negatively charged phosphate backbone with a neutral phosphorodiamidate linkage.
A neutral charge. Natural DNA is highly negatively charged. What happens when you remove the charge?
The molecule essentially becomes invisible to many cellular systems. Because it has no negative charge and a totally unnatural ring structure, blood proteins and nucleuses completely ignore it. It doesn't stick to anything, and it doesn't get degraded. It is incredibly stable.
Okay, but does it trigger the security guard?
No, because of its alien structure, it cannot trigger RNase H1, but it is absolutely perfect for steric blocking, acting as that immovable roadblock to alter splicing or block translation.
There has to be a trade-off. If it's invisible and indestructible, what's the downside?
The kidneys. Remember why the sulfur backbone was useful. It stuck to blood proteins, preventing kidney filtration. Because PMOs are neutral, they do not stick to albumin. They circulate entirely unattached.
And the kidneys just flush them.
And because they're relatively small, the kidneys filter them out of the blood incredibly rapidly, dumping them into the urine.
So you have a highly stable, non-toxic drug that gets flushed out of the body before it has a chance to enter the target tissues.
Yes, the clearance rate is extraordinarily high. To get enough of a PMO into the tissues to have a therapeutic effect, especially in tissues like skeletal muscle patients, require very frequent, massive intravenous doses. We're talking huge quantities of the drug administered weekly. It places a massive logistical and physical burden on the patient.
Which perfectly transitions us to the clinical reality. We've established the mechanisms shredding or blocking RNA. We've built the armored chemistry, the gapmers, the sulfur, the stealth PMOs. Let's look at where these custom molecules are actually being deployed right now. What diseases are we effectively targeting?
The most dramatic success story for ASOs so far has been in neurological disorders. The central nervous system is incredibly difficult to drug, but ASOs have a unique advantage when it comes to delivery.
The primary hurdle for the brain is the blood-brain barrier. It's an aggressive filter that prevents almost all traditional drugs circulating in the blood from entering the brain tissue.
The blood-brain barrier is famously impenetrable. But with ASOs, clinicians can bypass it entirely using an intrathecal injection.
A spinal tap.
Right. They inject the drug directly into the cerebrospinal fluid, usually through the lower back. The ASO circulates in the spinal fluid, bypassing the blood-brain barrier, and bathes the entire brain and spinal cord, getting taken up by the neurons.
And this delivery method led to the breakthrough for spinal muscular atrophy, or SMA. Let's explore SMA because the biological mechanics of this disease and the ASO that treats it are fascinating.
SMA is a devastating and historically fatal genetic disease in infants. It causes progressive muscle wasting because the motor neurons in the spinal cord die off. It's caused by a severe loss-of-function mutation in a gene called SMN1, which produces the survival motor neuron protein. Without that protein, the motor neurons die.
But if the gene is completely broken and the protein is missing, how does an ASO help? We said earlier, ASOs are good at stopping toxic proteins, not replacing missing ones.
This is where human genetics provides a backdoor. Humans happen to have a nearly identical backup gene called SMN2.
A backup generator.
The problem is SMN2 has a tiny single-letter defect that ruins its splicing. When the spliceosome tries to edit the SMN2 pre-mRNA, it accidentally skips a crucial exon 7. Because exon 7 is missing, the resulting protein is unstable and degrades almost immediately. It produces only a tiny fraction of functional protein.
So the backup generator is on, but the wiring is faulty.
Exactly. The ASO designed for SMA is a steric blocking molecule. It doesn't destroy anything. It is injected intrathecally, enters the motor neurons, and binds to a very specific spot on the SMN2 pre-mRNA and intronic splice silencer. It physically masks the chemical signal that was telling the spliceosome to skip exon 7.
It covers up the skip me sign.
Yes. By hiding the silencer, the ASO forces the spliceosome to include exon 7 in the final transcript. Suddenly, the backup SMN2 gene starts producing massive amounts of fully functional, stable motor neuron protein. It fundamentally rewires the RNA processing to replace the missing protein, transforming a fatal infantile disease into a manageable condition where children are meeting motor milestones.
That is just, it's an incredible application of the splice switching roadblock. They're also using intrathecal ASOs for ALS, right? Lou Gehrig's disease?
Yes, but using the opposite mechanism. In certain genetic forms of ALF, mutations in the SOD1 gene create a classic toxic gain-of-function protein that destroys motor neurons.
So they need the wood chipper.
For this, they use a gapmer ASO to recruit RNase H1. The ASO binds to the SOD1 transcript, shreds it, and shuts off the production of the toxic protein, slowing the neurodegeneration.
So the nervous system is highly amenable to ASOs via spinal injection. What about muscular disorders? We talked about PMOs and massive intravenous doses earlier.
Muscular disorders are the prime target for those neutrally charged PMOs. Duchenne muscular dystrophy, or DMD, is the hallmark application. DMD is a severe, progressive muscle wasting disease caused by mutations in the dystrophin gene, which is one of the largest genes in the human body.
And the size of the gene is part of the problem. You can't just deliver a replacement gene using a traditional viral vector because the dystrophin gene is physically too massive to fit inside a therapeutic virus.
Exactly. Gene replacement is largely off the table. In DMD, the mutations usually cause a frame shift. The genetic code is read in three-letter words. If a mutation deletes an exon that isn't a multiple of 3 letters, it throws the entire reading frame out of alignment.
Oh, I see.
Everything downstream becomes genetic gibberish, resulting in a premature stop signal. No functional dystrophin protein is made.
So the sentence reads, the dog ate the cat. If you delete the O in dog, it shifts everything over and it reads, the dia tet heck at. It's totally ruined. How does a PMO fix an out-of-alignment frame?
By strategically deleting more of the sentence.
What?
The PMO acts as a splice-switching roadblock. It binds to the pre-mRNA and forces the spliceosome to completely skip over a specific targeted exon adjacent to the mutation.
Wait, if the protein is already broken because it's missing pieces, how does skipping another piece help? Don't you just get a shorter, even more broken protein?
You do get a shorter protein. It is internally truncated. But by carefully selecting which exon to skip, the chemists restore the reading frame of the rest of the gene.
Oh.
You delete 2 words, but the sentence makes sense again. The resulting dystrophin protein is shorter than normal, but it is partially functional. This exon-skipping strategy converts a severe, fatal DMD phenotype into a much milder, slower-progressing form of muscular dystrophy.
That's a masterclass in making the best of a bad situation. Okay, we've covered the brain and the muscles. What about the liver? Earlier we mentioned the liver naturally absorbs these drugs.
The liver is the body's primary filter, meaning it is a massive sponge for oligonucleotides. If you inject a standard sulfur-modified ASO under the skin or into a vein, the liver naturally takes up the vast majority of it. But chemists weren't satisfied with natural uptake. They wanted surgical precision. They achieved this using GalNAc conjugation.
GalNAc. It's spelled G-A-L-N-A-C. Break down what that is.
GalNAc is a specific type of complex sugar molecule. Hepatocytes, the primary functional cells of the liver, have a high density of specialized receptors on their surface called ASGR. These receptors are specifically designed to look for and bind to GalNAc sugars floating in the blood.
So chemists use the sugar as molecular address label?
Exactly. They chemically tether a cluster of these GalNAc sugars to the end of the ASO. When the drug enters the bloodstream, it circulates until it hits the liver. The ASGR receptors grab the GalNAc sugars with incredible affinity and actively drag the attached ASO inside the cell through endocytosis.
It's a VIP pass straight into the hepatocyte.
The increase in delivery efficiency is astronomical. Because of GalNAc conjugation, developers can treat liver-mediated diseases like severe hypercholesterolemia or rare conditions like ATTR amyloidosis, where the liver pumps out toxic amyloid proteins at a tiny fraction of the dose they used to need. You lower the dose, you massively reduce the systemic toxicity, and you increase the potency.
I also noticed in the clinical reviews that ASO technology is beginning to cross over into oncology. How are we targeting cancer? Cancer isn't usually a single monogenic mutation.
Oncology is a complex frontier, but ASOs offer unique angles of attack. One fascinating application is acting as a telomerase inhibitor. Telomerase is an enzyme that many aggressive cancers use to endlessly rebuild the protective caps on the ends of their chromosomes, the telomeres. This essentially makes the cancer cells immortal. They can divide forever without their DNA degrading.
And an ASO can turn off the immortality switch?
Yes, but in a very unique way. The ASO doesn't target the protein part of the telomerase enzyme. Telomerase is a complex that actually contains its own built-in RNA template, which it uses to build the DNA caps. The ASO is designed to bind directly to the essential RNA component, completely shutting down the enzyme's structural capability. Without active telomerase, the malignant cell's telomeres rapidly shorten with each division, triggering crisis and apoptosis-programmed cell death.
The applications are incredibly diverse. We are treating the brain, rewiring muscles, reprogramming the liver, and attacking cancer. But let's take a step back into the reality of drug development. Before any of these custom-designed molecules are injected into a human being, how do we fundamentally prove they are safe? Because we established earlier that if an ASO is slightly too sticky or binds to the wrong target, it can wreck an organ.
The safety assessment pipeline for ASOs is uniquely rigorous and requires entirely different protocols than traditional small molecules. Regulatory agencies require a comprehensive focus on off-target effects. Remember, the core mechanism relies on a 20-letter sequence being entirely unique within a human genome that contains 3 billion letters.
Right. What if the 20-letter sequence we designed to hit the toxic Huntington's gene happens to perfectly match a sequence in a gene required for heart function? If we accidentally shred the heart gene, the cure is worse than the disease.
That is the nightmare scenario. To prevent it, developers employ a strict, 3-tiered off-target assessment protocol. Tier 1 is computational prediction. Long before a chemist ever synthesizes the physical drug, powerful algorithms scan the entire known human genome and transcriptome in silico. They're looking for sequences that are a perfect 100% match, but they also scan for near matches, sequences with 1, 2, or 3 mismatches.
Because if the chemistry is sticky enough, an ASO might bind to a sequence even if it's off by a letter or two. The computers are generating a massive hit list of every potential accident waiting to happen.
Exactly. They map out the theoretical danger zones. Tier 2 is transcriptomic experimental validation. The computers make the map, but biology is messy, so you have to test it in living cells. They take human cells grown in a dish, treat them with the actual physical ASO, and then use RNA sequencing. They literally sequence every single piece of RNA in the treated cell and compare it to an untreated control cell.
They are checking the computer's homework to see if any of those unintended targets on the hit list are actually being destroyed.
And often they find hits the computer missed, or realize that theoretical matches don't bind in reality because of how the RNA is folded. Finally, Tier 3 is RT-qPCR confirmation. For any unintended RNA targets that showed a drop in the RNA-seq data, the researchers use highly specific quantitative polymerase chain reaction tests to measure exactly how much of that specific RNA is being lost, verifying if it crosses a dangerous threshold.
It's an exhaustive 3-stage filter. And when these drugs finally make it through those tiers and enter clinical trials in actual human patients, how are they measuring success? They aren't just asking the patient if they feel better.
No, clinical endpoints are heavily driven by biomarker knockdown. Because ASOs act directly and predictably on RNA, you should be able to measure a physical reduction in the toxic protein in the patient's biofluids. For example, in those ALS trials we mentioned, they measure the reduction of neurofilament light chain in the blood or spinal fluid.
What is that?
Neurofilament is a structural protein in neurons. When neurons die, they spill it into the blood. By measuring its decline, they can literally track the cessation of nerve damage in real time.
It sounds incredibly clinical and foolproof, a neat sequence of steps. But anyone who follows medical science knows drug development is never that clean. Biology always fights back. What are the blind spots here? Where do things still go wrong with modern ASOs?
That leads us to the critical limitations and barriers that the field is actively struggling with today. The most glaring limitation, without a doubt, is delivery to extrahepatic tissues. Extrahepatic simply means anywhere outside the liver or the central nervous system.
Because the liver is a giant vacuum that sucks up all the systemically injected drug, and the brain has to be accessed through an invasive spinal tap.
Exactly. What if you want to treat a genetic heart disease? Or a disease deep in the skeletal muscle? Or the lungs? It is incredibly difficult to achieve a high enough therapeutic dose of an ASO inside those tissues without administering massive amounts of the drug. And if you administer massive amounts, the liver absorbs the vast majority of it, which brings us to toxicity.
And the literature reveals something really insidious about ASO toxicity. It's not always about off-target RNA binding, is it? It's not just accidentally cutting the wrong wire.
No. Some of the most severe toxicities are sequence independent. They have absolutely nothing to do with Watson-Crick base pairing or targeting the wrong RNA. They are driven entirely by the biophysical shape and chemistry of the ASO itself.
You mentioned this earlier with the LNAs, the locked sugars making the drug too sticky.
Highly modified, highly sticky ASOs, especially those with a high concentration of sulfur linkages and locked sugars, have a tendency to bind non-specifically to intracellular proteins. A major recognized culprit in ASO toxicity involves perispeckle proteins.
Perispeckles, it sounds microscopic. What are they?
They are tiny, dynamic subnuclear structures built out of RNA and specific proteins. They help regulate gene expression. When certain toxic ASOs enter the nucleus, they bind to these perispeckle proteins with massive affinity. They bind so tightly that they physically drag the proteins out of their normal position, pulling them into a different structure called the nucleolus.
So the ASO acts like a molecular wrecking ball. It doesn't even bother hitting the wrong RNA. It just tackles a vital structural protein and physically relocates it across the nucleus.
The disruption is profound. This dragging causes massive nucleolar stress. The cell immediately senses that its internal architecture is collapsing, halts all functions, and triggers apoptosis. The cell hits its own self-destruct button.
And if this is happening on a macro scale, what does it look like for the patient? What organs take the hit?
Historically, it manifests as severe organ toxicity. Hepatotoxicity liver damage is the most common because the liver absorbs the lion's share of the drug, so its cells experience the highest concentrations. Nephrotoxicity, or kidney damage, is also a major hurdle. Even though the ASOs bind to blood proteins, they eventually break down and have to be cleared. They accumulate in the proximal tubular epithelial cells of the kidneys, causing cellular stress and damage.
The safety reviews also highlight issues with blood clotting.
Thrombocytopenia, a dramatic, sometimes sudden drop in blood platelets, which are essential for clotting. This can lead to severe bleeding events. It was a major issue with some early-generation ASOs, likely triggered by complex sequence-independent interactions between the highly modified backbones and the immune system or platelet precursor cells.
And compounding all of this is the fact that these drugs are not one-and-done cures.
They are chronic therapies. ASOs are eventually degraded by nucleuses or cleared from the body. Depending on the chemistry, repeated dosing is required, perhaps monthly, perhaps every 3 to 6 months. Every time you introduce a highly modified synthetic molecule into the human body, you raise the cumulative risk of long-term immune activation, subtle inflammatory responses, or accumulating organ toxicity.
So we have to be incredibly clear. ASOs are not a magic bullet. They operate within a very narrow therapeutic window.
That is the precise term, a narrow therapeutic window. The gap between the dose required to silence the disease and the dose that causes unacceptable toxicity can be very tight. ASOs are not inherently dangerous, but their safety is a delicate high-wire act, highly dependent on balancing the exact chemical backbone, the sugar modifications, the sequence, and the dosing schedule.
We have covered massive ground here. We've gone from the brutal.
Yes. Developers are taking the highly potent ASO and covalently linking it to a monoclonal antibody. The antibody is engineered to seek out and bind to very specific receptors that are highly expressed on the surface of hard-to-reach tissues. For example, they are attaching ASOs to antibodies that specifically target the transferrin receptor, which is abundant on the surface of muscle cells.
So the antibody acts as a microscopic transport vehicle. It drives through the bloodstream, ignores the liver entirely, finds the muscle cell, locks onto the transferrin receptor, and drops the ASO payload directly inside.
It acts as an incredibly precise homing beacon. By shuttling the ASOs directly into the muscle or heart tissue, developers believe they could achieve massive, transformative gene silencing at a fraction of the systemic dose. If it works consistently, it solves the delivery limitation and drastically widens that narrow therapeutic window by bypassing the organs vulnerable to toxicity.
That is a brilliant engineering solution. But there is another trajectory mentioned in the literature that feels less like a biological breakthrough and more like a philosophical shift in how we approach medicine entirely. Personalized RNA therapeutics, N-of-1 trials.
This is perhaps the most profound capability of the ASO platform, because ASOs are fundamentally just programmable strings of code software that you can design and synthesize on a machine in a matter of weeks. They possess the unique ability to be rapidly customized for ultra-rare diseases.
We are talking about nano-rare diseases, genetic mutations so unique that they might only affect a handful of children or even just a single child in the entire world.
Historically, the pharmaceutical model is driven by scale. It cost billions of dollars and a decade of research to develop a small molecule drug. You cannot justify that investment for a disease that affects one person. Those patients were entirely orphaned by modern medicine. But ASOs shatter that paradigm. If a geneticist sequences a child's genome and discovers a totally unique, fatal splice site mutation, researchers can design a custom ASO sequence to mask that specific mutation. They can synthesize it, test it in the patient's own cells in a lab, prove it fixes the splicing, and manufacture a clinical-grade batch in a matter of months.
This isn't theoretical. There was a landmark case that proved this model, the story of Malaysin.
Yes, Malaysin was a bespoke ASO drug, literally named after the little girl it was designed to treat, Mila. She had a unique, rapidly progressing genetic disorder caused by an aberrant splicing mutation. A team at Boston Children's Hospital designed, tested, and received FDA permission to administer a custom ASO in under a year.
Less than 12 months from discovering the mutation to infusing a custom-built programmable genetic drug.
It was unprecedented. The drug successfully corrected the splicing defect and dramatically reduced her severe, life-threatening seizures. While it couldn't reverse the damage already done, it fundamentally proved that an N-of-1 drug, a therapy designed and manufactured for exactly one human being on the planet, is a viable scientific reality.
That stretches the definition of what a pharmaceutical is. But the innovation isn't stopping at just silencing RNA or acting as a splice-blocking roadblock. What is the next generation of this tech?
The horizon is moving from RNA silencing to active RNA editing. We are seeing the rise of a modality called ADAR recruiting ASOs, frequently referred to as AIMers.
ADAR. ADAR. Let's define the acronym.
ADAR stands for adenosine deaminases, acting on RNA. Like RNase H1, ADAR is an endogenous, naturally occurring enzyme floating in your cells, but it doesn't shred RNA. ADAR is a chemical editor. It has the ability to chemically convert an adenosine, the A, in the genetic code into an inosine. And the cellular machinery reads an inosine as a guanine, or a G.
It's a natural enzyme that can actively erase an A and write a G.
Exactly. AIMers are cleverly designed, highly structured ASOs. They bind to the mutated RNA sequence, but instead of triggering destruction, their unique physical shape actively recruits this ADAR enzyme, pulling it right to the exact location of the mutation. The enzyme executes the chemical edit on the fly, physically changing the mutant letter back to the healthy letter.
So you aren't destroying the transcript. You aren't forcing the spliceosome to skip exons and build truncated proteins. You are using the cell's own spell checker to erase the typo in the photocopy. And because it's only editing the transient RNA messenger, you aren't permanently hacking the patient's master DNA genome.
It is the ultimate transient precise correction. It repairs the transcript temporarily, lowering the risk of the permanent, irreversible off-target edits that remain a concern with DNA editing technologies like CRISPR. If you start taking the ASO, the editing stops. It provides a level of control that is incredibly appealing to regulatory agencies.
This entire exploration has been staggering. We started with an audacious 13 nucleotide experiment in 1978. We waded through a brutal trough of disillusionment where the human immune system and blood enzymes shredded every attempt to make this work. We explored the meticulous atom-by-atom chemical engineering, the sulfur swaps, the locked molecular bridges, the gapmer cookies that armored these fragile molecules. And now we are looking at a mature, multimodal, precision medicine platform capable of reaching into the brain, rewiring muscle genetics, and creating entirely bespoke cures for a single individual.
It is a profound testament to the perseverance of medicinal chemists, molecular biologists, and the patients in those early trials. They took a concept that was biologically elegant but chemically impossible, and through sheer iterative force, bent it into a therapeutic reality.
Which leaves us with a final concept to mull over. We've highlighted how this technology allows for an N of 1 drug, a completely bespoke therapy designed, synthesized, and approved for exactly one human being. But as this platform scales, it forces us to ask a massive structural question. If medicine is no longer a mass-produced chemical in a factory, but rather a software patch, written in a lab for your specific unique genetic code, how does the global healthcare system adapt to that? When the line between a traditional pharmaceutical drug and a string of custom code blurs entirely, how do regulatory agencies, insurance models, and hospitals ensure that everyone gets the biological update they desperately need? Thank you for joining us on this deep dive. Keep questioning the world around you.